So let's review some key concepts. We learned that primary tumor cells are encouraged by the reactive stroma. That means the surrounding stroma that was supporting the normal epithelial cell growth becomes manipulated due to many mutations in the epithelial cells that then stumble upon different types of signals that begin to alter the way the stroma communicates with it. And this reactive stroma then supports or even encourages the ability of these epithelial cells to begin to move through mesenchymal tissues. So this is the stromal tissues, and get into places like nearby capillaries, that process of intravasation or entering the vasculature. We also learned that macrophages are recruited as part of the wound healing response that the mutating tumor cells have stumbled upon. And the actions of these macrophages then ease the way for the tumor cells to undergo this EMT, or epithelial mesenchymal transition. One of the sort of markers or changes that happen in this EMT process is that the epithelial cells stop producing certain types of cell surface receptors that are normally used to keep the healthy epithelial cell in place, nudged up against other epithelial cells. So proteins like E-cadherins get shut down, which then encourages the epithelial cell to separate from its neighbors. And as we discussed last time, this can also coincide with improper activation or expression of different types of cadherins, like N-cadherins, which are used by stromal cells to talk to each other. So then you suddenly have epithelial cells that want to mingle and talk, quote unquote, with other stromal cells, and that facilitates these epithelial cells moving through them. We ended last time discussing this concept that in order for circulating tumor cells in the blood that go through a lot of hardship and challenges in the blood, because this is not what they were necessarily designed for, if they end up stuck somewhere in a capillary, in some other tissue in another part of the body, in order for them to begin to colonize and really become disseminated tumor cells that are now lodged in other tissues, this will require them to revert back from the sort of newly gained mesenchymal type of behavior back to their original epithelial behavior, so that they can resume growing in this dysfunctional kind of tumor formation at a new site. And we say that the reverse of EMT is MET. So one thing to think about is how much of this metastatic phenotype, the cancer getting to the point where it's spreading throughout the body, is actually due to the tumor cell's own messed up genotype versus the encouragement of surrounding tissue. So now we are very much into this idea of a dialogue, right? How much of it is the messed up genome of the tumor cell? How much of it is contributed by the stroma surrounding it? So in other words, to go back to this seed and soil analogy, which I shared with you as kind of a classic way of thinking about how tumor cells that leave a primary tumor site and circulate around, how it is that it's actually unlikely that they end up somewhere in the body that's conducive for them to form another tumor. And we said it's like a seed being scattered around in different types of soil, and you are not surprised that in only under certain soil conditions will these seeds grow, right? In other conditions, the seeds just sit there and do nothing. So if we think about this, how much of it is actually the genotype, and how much of it is the good soil? Let's take a look at some further examples of how other cells are contributing to this metastatic phenotype. So here we have a cartoon depicting a primary tumor that is showing that the basement membrane has broken down because these cells are recruiting macrophages, perhaps as part of the wound response. So these macrophages, represented in yellow, they were just maybe circulating through the blood as monocytes. And then when they got to the site, they began to pick up some signals that are encouraging them to undergo diapedesis. Remember, that's normally the term for things like white blood cells leaving the blood, squeezing between the walls of the capillary and getting into the stromal tissue. Right? And at this point now, we call them TAMs, or tumor-associated macrophages. Right? So now these tumor-associated macrophages continuously are stimulated by not only the wound healing response, but by additional types of signals that the tumor cells have now learned how to send out as part of the random mutations that they're undergoing. One of these is colony stimulating factor, or CSF1. CSF1 is the signal that recruits macrophages and really encourages the macrophages to come to where the tumor is. So CSF1 is a signal that encourages monocyte differentiation into macrophages. So the monocytes turn into macrophages, and they also divide, multiply, and become more active. At the same time, these macrophages, because they are receiving this CSF1 signal and are stimulated to divide and proliferate, they are communicating with each other by using epidermal growth factors, or EGF, to stimulate each other to divide. So not surprisingly, these EGF signals can also be picked up by the cancer cells and used improperly to stimulate even more aggressive tumor cell division. So this is a paracrine signaling loop, back and forth. We can see that depicted on the cartoon here, this back and forth of EGF and CSF, and we can certainly see it in this Western blotting data. So here we are looking at, if you were to go ahead and actually look at the protein content of macrophages that are being recruited to a tumor, as well as the carcinoma cells of the tumor itself. So beta-actin protein levels just up here are a loading control to show you that you're looking at the same amount, relatively speaking, of macrophages versus carcinoma cells in terms of the amount of total protein that's coming out of the cell. But then you can compare and actually see what different levels of proteins are in that, are in these cells. So EGF receptor, you can see that the macrophages, right now are not expressing a lot of EGF receptor, but the carcinoma cells actually start expressing a lot of this EGF receptor to receive EGF. The macrophages are the ones producing the EGF. So you can see here, this is EGF protein, right? So it's not the carcinoma cells making EGF, but the carcinoma cells can certainly respond to the EGF because they're upregulating EGF receptor. Conversely, the macrophages have on their surface CSF1 receptor, and it's the carcinoma cells that have now improperly learned or stumbled upon making a lot of CSF. So this is what we mean by there's this very closely knit heterotypic signaling going on between these two different cell types that unfortunately in this case leads to more aggressive metastases. How important is this CSF1 in terms of recruiting macrophages, and how important is that recruitment of macrophages to the aggressiveness of the tumor? You can look, here are some lab results. These are looking at tumors in mice that are engineered to be CSF negative. In other words, their macrophages, they are not able to go ahead and receive CSF signal. So the CSF gene has been knocked out. If you look at wild type mice, and here are the tumor cells inside the wild type mice. So these areas up here are the tumors still sort of contained together, the cells. Here you can see is a region where the basement membrane has broken and the tumor cells are now making their way into the stroma. So in here is a mix of invading tumor cells, and most likely many of these cells in here are also macrophages or TAMs that have come and are actually remodeling the stroma to encourage the tumor cells to come out. On the right are the same tumor type, but in mice who are engineered to be CSF knockouts. So they don't have this CSF signaling between the cancer cells and the macrophages. And you can see very clearly there's little evidence of macrophages in the surrounding stroma being recruited here. And likewise, there's very little evidence of the breakdown of the basement membrane so that the cancer cells are still contained in situ. Another type of growth factor is hepatocyte growth factor, or HGF. HGF also stimulates invasion because HGF encourages epithelial cells to lose their dependence on sticking to each other. Right? And so if you lose your dependence on sticking to each other, that encourages the spreading out of the cancer cells. On the left here, so these are cells grown in culture, in tissue culture in the lab. So this is normal medium. They're grown in there. These are the cancer cells. And these are epithelial cells. So they are stuck together because normally that's what they want to do. If you were to grow them in three-dimensional cultures, meaning instead of on a flat surface of plastic, you were to sort of embed these cells in a matrix of some sort of a gel that is soaked with media and things like that, then the cells can start growing sort of in three-dimensional space. They would still very much stay together as balls, right? So these are balls of cells. However, if you go ahead and treat these cells with high levels of this hepatocyte growth factor, now keep in mind, this is yet another example of an important, widely used growth factor by many cell types that were originally, this growth factor was originally identified in liver cells, right? So that's why they're called hepatocyte growth factors, but the name is a little misleading because it can be used by many different cell types. If you give these epithelial cells this HGF, you can see now these epithelial cells spread out from each other. And in the 3D culture, instead of staying as little balls of cells, right, they're actually free to move around. And you can see what's happening here is they're actually spreading in three-dimensional space. And you can really imagine this being something like a tumor that's beginning to grow and spread. Certain transcription factors inside of the cell are very important for this whole epithelial to mesenchymal transition. When we think about different instances where epithelial cells should be encouraged to let go of each other and migrate through stroma, we've already discussed how in the process of wound healing, that is a very desirable sort of program of different changes that the epithelial cells should undergo. But if you go way back in time to the early embryonic development of, let's say, a person, right, or any organism, multicellular organism, you also imagine that as part of the newly formed blastocyst and newly developing embryo, of course, when you only have basically a ball of totipotent stem cells, as they begin to form those necessary 3 germ layers early on, that's going to require a lot of cells migrating and being encouraged to migrate. So key important transcription factors that are used in early development of the embryo include these 3 important ones. They have funny names: snail, slug, and twist. We can look at the expression levels of these 3 very important early developmental transcription factors in these examples here. So this is a cross-section of early embryo of Amphioxus. So this is a primitive, very primitive fish. And you can see early on, you have expression of the snail transcription factor in these cells here. And expression of snail in those cells encourage those cells to begin to form things like the neural crest. You can see very clearly this is not random expression. This is expression taking place, encouraging the migration of these cells to form the proper formation, for example, of the spine. Here is expression in early frog embryo of the slug transcription factor. And again, you can see it's helping to define where the spinal cord is going to be. So the epithelial cells or the developing cells of the germ layers are migrating based upon their expression of slug. Lastly, in flies over here, this is Drosophila. This is expression of twist. So in this early fly embryo, where there's maybe only a few hundred cells, you can see that these cells on the dorsal side of the fly's abdomen are expressing twist. And twist then encourages these cells to migrate and begin to form this structure that's going to run up and down the dorsal part of the fly embryo. So snail, slug, and twist then, we have the same transcription factors. We have homologs, our human versions of snail, slug, and twist that we used in our early embryonic development as well to carry out these very same functions. So they are expressed, of course, by their corresponding genes. And remember, these genes still exist in every nucleated cell in your body, right? But because now their function in terms of early embryonic development has long since been turned off, right? Because you didn't need it anymore. Now that you're in an adult body, that means these three genes are heavily methylated to keep them shut down in the different cell types in your body now, because you don't need them anymore. The exception is when you are injured. If you have a cut and then the wound healing response kicks in, then these genes become hypomethylated, or the methylation is reversed. So in those epithelial cells, they may switch on again, snail, slug, and twist, in order to then be part of this epithelial to mesenchymal transition. So in adults, that means that these EMT transcription factors are only transiently expressed when needed, specifically when wound healing takes place. So here we have in tissue culture, another tissue culture experiment, where we've looked at this type of experiment before, where people who study wound healing oftentimes will grow a nice uniform layer of epithelial cells in the dish, and then here scrape off some of these cells. And this kind of represents, like a wound. And you can see here that the slug protein is being labeled in brown. So all of these normal epithelial cells growing here, they of course have the slug gene in them, because it's not like they magically lose the gene. The gene is just shut down. Right? But when needed, the gene is woken up again. And so you can see the cells that are right next to the edge of the wound begin expressing slug. And then after 48 hours, you can see the cells that are migrating are expressing heavy levels of slug. Right? After 72 hours, this begins to subside. And after 96 hours, once the area has been filled in, then they shut the slug expression off again. So what we're looking at then is very much the expression of these transcription factors being tightly controlled and expressed transiently. So normally, how is it that these transcription factors are causing these changes? So normally, snail, slug, and twist, right, when they're shut down, they don't interfere with anything in the cell. But when they are expressed, they actually discourage the epithelial cells to express E-cadherin. So once the epithelial cells stop expressing E-cadherin on the surface, that's how they begin to lose their attachment to their neighboring epithelial cells, right, to encourage them to feel comfortable moving through the stroma. So remember that these EMT transcription factor genes are normally active in early embryo, but they're silenced in our adult cells except for brief episodes when you need to switch them on and they can do their thing. Consider then that improperly activating these transcription factors, such as cancer cells undergoing random mutations that may end up resulting in the removal or the reversal of methylation of these transcription factor genes, might be another way that cancer cells gain a metastatic advantage. And this just down here is just to help remind you that what we're looking at in these images is epithelial cells undergoing EMT to become more mesenchymal, right? And then going the reverse, MET to become epithelial again. So these EMT transcription factors, it's well established now that one of the properties they give cells is sort of this reversion to becoming more like a stem cell again. So if we look at the expression here, these are HMLE, which is a mammary epithelial cell line. It's not necessarily a cancer cell line, but it's a model of normal epithelial cells. So normally they would be sticking together because of their E-cadherin expression and their normal dependence on being attached to each other in order to function properly as an epithelial cell. But if you give them a lot of, or you overexpress a lot of the snail transcription factor in them, you can see very clearly their behavior changes. They're much happier being spread out now. If you look at different types of markers on the surface of these cells, such as CD44, which is normally associated with stem cells, you can see that the normal epithelial cells, when they're growing together, the bulk of them, right, do not have much CD44 expression. These would be considered the non-stem cells, where you'd have a subset of them, right? Maybe the few that are sort of representing things like stem cells would have the CD44 marker. But if you overexpress snail in them, an interesting thing happens. Not only will they spread out, but they begin to take on more of this stem cell-like marker on their surface. Okay. You can do that with overexpressing snail. You can do that by overexpressing twist as well. And you can see you dramatically shift the apparent behavior of these epithelial cells from being epithelial to being more stem cell-like. I ask up here, why would this normally be used in embryogenesis? Well, because you imagine, if you only have a few hundred cells at a particular early point in embryonic development, and all of those cells are eventually going to go on through division to contribute to billions, if not trillions of cells that make up the body, then obviously early on, these all represent very, very pluripotent stem cells. Right? So during early embryogenesis, the actions of snail, twist, and slug, right, maintain that high pluripotent stem cell-like state. If we look at the effect of these transcription factors in vivo, so these are experiments where you can actually see if you go ahead and transplant some epithelial cells to new stroma, and then you encourage them by expressing things like slug, for example, you can see what happens. So here, for example, is a part of a stromal fat pad. So in other words, this would be from breast tissue. This would represent the stroma that normally supports the epithelial cells that make things like the ducts and the glands. So you can remove, let's say, the ducts that are in there, leaving just kind of this very clean stromal tissue. And then you go ahead and you implant test cells in there. In this type of environment, then, you wait and see if these implanted test cells will actually form new ducts, if they will be stimulated to go ahead and spread and actually grow. So in this data down here, we're looking at these mammary epithelial cells under control conditions when these cells are just put into the stromal fat pad with no additional encouragement, not much growth happens. Right? But if you go ahead and you put, let's say, 100 of these MEC cells into the stromal pad, and then you have them express SOX and slug and other transcription factors that are part of their growth proliferation, such as SOX9, you can see you get very dramatic development of new tissue. So I always like relating these things that we talk about back to the clinic, the oncology clinic, and how these different things that we talk about as being potential markers, right, are they in real life used as diagnostic or prognostic tools? So what about these early embryonic transcription factors, right, like snail, twist, and slug? In fact, we have seen that in the clinical setting, patients who present with tumors, when they originally have their biopsies done and histology is done to kind of look at what proteins these are expressing, we can see that these early developmental transcription factors serve as a very useful clinical marker. Up here, So we're looking at 3 Kaplan-Meier plots now. On the y-axis is a measure of how many patients in this cohort that are being followed, how many years they can go on after treatment before relapse of their cancer. Right? And so on the x-axis is the number of years following the initial start of being followed here. And you can see patients who their biopsy of their tumor showed very little expression of snail, for example, right? Their survival is in green here. Whereas patients whose tumors, when they were first biopsied, the pathologist was able to detect high levels of snail protein inside these cells. The red, right, is their prognosis, right? Big difference. And so this is seen as well if you look at other EMT transcription factors like twist, right? So high twist versus low twist, the difference in survival rate of the patient. And when you look at slug, so in this case, this study was just looking at whether or not they could detect slug or not in the cells, right? So minus slug versus plus slug. So very clear that the awakening of these transcription factors as part of the many, many different mutations that are possible for the cancer cells to stumble upon, right? These are very important ones as well. We also want to take a look at what the cancer cell itself is doing to the surrounding stroma as it's moving through. We already discussed that part of the epithelial cell wanting to leave its other epithelial cell neighbors and go through the stroma. Part of it is losing E-cadherin expression and maybe gaining N-cadherin expression. Maybe there are not transcription factors, but maybe there are growth factors like HGF that also encourage the epithelial cells to separate. But then when the cell is actually in the stroma, what is really enabling the cancer cell to make its way through it? Right? It's one thing to say, well, they are now more mesenchymal so that they are okay in the stroma, but what's actually allowing them or encouraging them to move? Right? There are certain classes of matrix metalloproteinases that act on the surrounding stroma that are secreted by the cancer cells themselves. An important group of them are these membrane type MMPs. And these are used by cancer cells to carve their way through the extracellular matrix. Here we see a cancer cell, and this represents, let's say, the basement membrane. And the pink is the stroma beneath. So if the cancer cell stumbles upon mutations that wake up the expression of membrane type 1 MMP, this is actually a protease that's bound to the surface of the cancer cell, represented by these little red Pac-Men here that are still attached to the cell. So these proteases actually help eat away the surrounding basement membrane and stroma, just like the MMP enzymes that are secreted by the macrophages on the other side of the basement membrane. So that's part of how the cancer cell eats its way through the membrane by digesting proteins. Also, within the stroma, you'll have a lot of matrix metalloproteins that are still in their proenzyme form, meaning they're not activated yet. So these surface-bound MMPs on the cancer cell can actually activate these blue MMPs that are floating around in the stroma, right? And so then they become these blue Pac-Men. And together then, they're working to really digest and eat away all of these fibrous proteins that are in the stroma that might be blocking the cancer cell from making its way. And as you can see in this panel of the cartoon here, you effectively get the cancer cell carving a channel through the tissue. So here we can see how important these membrane type proteases are. So this is a tumor that's growing in a three-dimensional culture again, right? So you can actually see it's spreading out in 3 dimensions. And this has wild type MMT or MMT1 on the surface of the cancer cells. So as a result, you can actually appreciate how these cancer cells are sort of carving their way through the surrounding stroma. if this same tumor cells were engineered to remove their ability to express this MT1 on their surface, you can see a dramatic reduction in their ability to carve their way through the surrounding stroma. Very dramatic. And here, this is a wonderful image. So this electron micrograph actually shows a number of cancer cells in pink here, carving a channel through the stroma. So here, the blue, the light blue, are collagen fibers, right? Fibers that are part of the stromal network. And you can really imagine here that these cancer cells are basically digesting their way through, because on their surface are these MT1 proteases that are really beginning to cut and cleave these collagen fibers, digesting their way through. So here are some take-home messages at this point before we move on to the last couple of slides here. So it's sort of a summary. So we've been thinking about how heterotypic signals from the stroma are very important for invasion to happen. So in other words, just the cancer cell undergoing random mutations in its own sort of dysfunctional way of losing control of its cell cycle is not enough, right? The buildup of mutations and changes in the cancer cell itself are not enough to allow it to begin to migrate and move. It depends upon the involvement of this reactive, manipulated stroma around it. Number two, these EMT events, the epithelial to mesenchymal transition, it does not depend upon a whole sequence of haphazard mutations to happen, right? Because all you need are certain mutations that switch on a master program for these EMT transcription factors like slug, snail, and twist to be reactivated and re-expressed in the epithelial cell where they're normally shut down, right? And third, because the invasion is driven by the reactive stroma at the primary tumor site, and that's a big part of why the tumor cells have become very sort of aggressive and have now been encouraged or want to leave their primary tumor site. Once these motile cells that are floating around in our circulation, our CTCs, circulating tumor cells, once they've metastasized to a new home with normal, sort of quiet, mellow stroma, right, there's going to be a tendency for these epithelial cells to need to revert and undergo MET to get back to an epithelial form in order for them to really start then beginning to grow again into the tumor type that they once were back at the primary tumor site. So the last couple slides, let's just talk about this idea of what makes good soil for the seeds that are scattered in there. It's very clear that much of this depends on the genotype of the original tumor cell, right, and its own makeup of what it can and cannot sort of deal with in terms of being in a new home. And a lot of it deals with the target destination tissue, right, the metastatic site, and what the stroma in that particular tissue is used to doing in terms of signaling and communicating with epithelial cells. And so I think it's very interesting to think, again, that because we have many different tissue types in our body, right, and many different types of cells in those tissues, that yeah, this is an interesting combination of two things. So that's why not all tumor types metastasize to all different types of tissues in the body. This cartoon really highlights that. What they're showing are four different cancer types: prostate, pancreas, breast, and colon, and four possible destination tissues: brain, lungs, liver, bone marrow. And the size of the arrows, because the arrows represent metastases happening here, the size of the arrows give you an understanding or an appreciation for how frequent that particular metastases takes place. So in other words, prostate cancers, it is most common that they metastasize to bone marrow. It is least common that prostate cancer cells will metastasize to lung. Remember, this doesn't mean that the circulating prostate cancer cells that have come off the prostate tumor are not actually finding their way to all these tissues, because most certainly they're getting stuck in the lungs, just like they're getting stuck in the bone marrow. But this is the seed and soil thing, right? The ones that end up in the lungs don't grow because it's the wrong soil conditions. The ones that end up in the bone marrow, when the seed is scattered in that kind of soil, they grow very well. Pancreas, you can see very little pancreatic cancers metastasize to lung. A majority of pancreatic cancers will metastasize to liver. Breast can go to lung and bone, not so much to liver, but sometimes to brain, right? So these arrows exist, but they're smaller. Colon most frequently metastasizes to liver. So let's take a look at bone marrow, right? Because what is it about bone marrow that makes it such a fertile kind of soil for things like prostate and breast cells to be able to metastasize in? So bone is an interesting tissue because by its nature, bone is constantly remodeling. There is abundant cellular turnover in bone. And this involves two types of bone cells, the osteoclasts that go around digesting old bone so that all the minerals and nutrients can be reabsorbed and recycled, and the osteoblasts that then use those raw materials to lay down a new mineralized matrix. Many of you have probably at some point in your life suffered a broken bone, right? And you're well aware that our bones are able to heal, right? We usually don't think about our bones breaking unless you really do have a big break, like your ankle, leg, or arm, collarbone, whatever. But what you don't realize is that every single day of your life, your bones are suffering from numerous microfractures, especially the bones of things like your feet. Just by going for a walk, right? The constant pressure on them is going to develop many microscopic little cracks. All the time, these little cracks are being healed up by the actions of osteoclasts and osteoblasts. When you think about something like in civil engineering and you have a big bridge made out of concrete, sometimes in history, large bridges have collapsed and fallen down, right? And then they realize, oh, there was a series of cracks that had formed over time that eventually led to the failure of that bridge, right? It's not like the bridge had no cracks at all and suddenly fell down. It's the buildup of cracks. So that's the same thing with the bones in our body. Every day we get numerous, thousands of little cracks in our bone, but they're healed up. So that's why the bone is a constant place of remodeling. So here we have on the left a nice image. These purple cells on the bottom are the osteoclasts. They are moving upward here, digesting or eating away this material. This material in the middle is the mineralized bone, right? That's solid bone. This is a really neat image because you can really imagine them sort of munching their way up here, breaking down the bone, right? At the same time, you have osteoblasts. So these osteoblasts are developing over here. So I think actually this may actually be an osteoclast still on the top because it's digesting away the mineralized bone layer here and revealing underneath the bone the extracellular matrix beneath. And this extracellular matrix is very rich in growth factors. So that makes it a very fertile soil for things like the new bone to grow, so the osteoblasts to work and proliferate, and not to mention the bone marrow that's contained inside of your bones, right? There's lots of growth factors in there because it's a very rich hematopoietic environment where you have stem cells that are constantly making things like new blood cells. So this means that the fertile soil of the bone is a good spot for disseminated tumor cells to be able to find very rich growth factors in a conducive environment to begin growing. So then we can shift our focus and say, if we say the bone is a good target, well, then let's look at the actual cancer cell that has ended up in the bone. Because now we can say, how does the genotype of that cancer cell, once it's in that fertile soil, how is that going to affect, sort of in a dialogue between the stroma and the cancer cell, the outcome of what the metastases looks like? This idea of the seed itself having some influence on the soil, right, kind of gets away from our very strict classic analogy of spreading seeds onto different soil and waiting for wildflowers to grow, because you don't really anticipate that the seeds themselves have any impact on the soil that they land on. But of course, that's not the case in metastatic cancer, because the messed up genotype of the cancer cell, right, will start trying to manipulate and convince the new stroma to help support it. So in these three panels is a great example of this. Here on the left is normal bone. So in normal bone, you can see here the orange is the mineralized bone. So that's the hard calcified bone. And within the pockets or cavities within the bone, you would have your bone marrow, right? The dark purple is where you have all this rich growth factors, all these brand new cells that are being encouraged to grow and proliferate. Surrounding it on the outside is muscle, right? So you have some muscle up here, skeletal muscle here on the other side. So keep in mind this balance of mineralized bone to bone marrow. If you look at breast cancer and how breast cancer behaves when it ends up in bone, you can see what happens is that because the breast cells have certain sort of expectations of what they need the stroma to do, right? They manipulate the stroma, and now you see you get very little mineralized bone left and far more of this soft, kind of fatty tumor tissue inside. Right? This is why I think many of you are aware that part of breast cancer and late-stage breast cancer, patients often develop very brittle bones. Right? So that's one of the leading causes of injury in people who have breast cancer is the weakening of their bones, right? Because there's less emphasis now on mineralization and much more emphasis on the tumor kind of just spreading in the sort of soft marrow. On the other hand, prostate cancer cells, when they end up in bone marrow, they do the opposite. They actually encourage much more activity of the osteoblasts. So the osteoblasts begin to lay down a lot more mineralized bone. Right? So it's interesting to think that in this case, the soil in each of these cases started off the same. It's fertile soil, right? The bone tissue and the bone marrow. And so now what we're talking about then is the genotype of the cancer cell arriving there, and then engaging in this now manipulative dialogue with the new stroma, resulting in different presentations or different types of symptoms of different cancers. Okay, let's go ahead and stop there.